Welcome to this lecture on Teacher on Demand series. This series is pertaining to carbohydrate metabolism and we'll be discussing glycolysis, the basic reaction. Glycolytic pathway is also known as the Embden-Meyerhoff-Parnas pathway. It is the basic pathway for glucose where it is progressively broken down to produce energy. Unlike the TCA cycle, glycolysis occurs obligate in all the cells, and this reaction occurs predominantly in the cytoplasm. The biological significance of this reaction is that this reaction generates energy from glucose. It has important intermediate. It gives important intermediates like glyceraldehyde 3-phosphate and pyruvate, which have tremendous amount of metabolic significance. Another important use of this pathway is that a substantial portion of it is utilized in the reverse direction during the process of gluconeogenesis, that is, production of glucose from non-carbohydrate source. So the basic significance is that it is the first step where glucose is gradually broken down to produce energy. It also provides important intermediates like glyceraldehyde 3-phosphate, which will be utilized in formation of triacylglycerol, and pyruvate. Pyruvate has got many metabolic fate, and also a substantial portion of this reaction is used in gluconeogenesis. Now the basic reaction, in a previous video I discussed about the role of hexokinase and glucokinase and how glucose is trapped inside. The glucose enters the cell through the various glucose transporters or glut, and once it enters in the cell, it has to be trapped within the cell. This is mediated by the enzyme hexokinase, and that is a requirement of an ATP. Glucose 6-phosphate then undergoes an isomerization state to form fructose 6-phosphate. Fructose 6-phosphate is then acted by an enzyme phosphofructokinase. An ATP molecule is used to produce fructose 1,6-bisphosphate. In these two steps, we are giving ATP and converting glucose into an intermediate which can be used for subsequent reactions. This fructose 1,6-bisphosphate is acted upon by an enzyme aldolase and produces two intermediates, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Glyceraldehyde 3-phosphate and dihydroxyacetone phosphate are the two basic carbohydrate trioses in the body, and with an enzyme triose phosphate isomerase they can be interchanged. The further metabolic role is present in glyceraldehyde 3-phosphate, which we shall discuss in the next slide. Glyceraldehyde 3-phosphate then is acted upon by the enzyme glyceraldehyde 3-phosphate dehydrogenase. Here NAD is converted to NADH to form 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate is then acted upon by the enzyme phosphoglycerate kinase to produce 3-phosphoglycerate. During this reaction, a high energy bond is produced. ADP is converted to ATP. This is an example of a substrate level phosphorylation. The NADH which is produced in the previous step will then be transported to the electron transport chain to produce further energy. This is an example of a substrate level phosphorylation where ATP is readily produced. The 3-phosphoglycerate then undergoes a isomerization to form 2-phosphoglycerate. The 2-phosphoglycerate then, with the help of the enzyme enolase, loses a water molecule to form phosphoenol pyruvate. Phosphoenol pyruvate is then acted upon by the last enzyme of glycolysis to produce the enzyme, that's pyruvate kinase, to produce an intermediate pyruvate. In this also another level of substrate level phosphorylation occurs. So if you have a look at glyceraldehyde 3-phosphate, conversion of glyceraldehyde 3-phosphate to pyruvate involves production of 1 NADH. This 1 NADH will go to the electron transport chain to produce 2.5 ATPs, and there are two levels of substrate level phosphorylation, that is catalyzed by the enzyme phosphoglycerate kinase and pyruvate kinase. Getting down to the energetics, in the first step where glucose is converted to glucose 6-phosphate and fructose 6-phosphate is converted to fructose 1,6-bisphosphate, two ATP molecules are utilized to produce energy, so we are using two high energy bonds. Subsequently, when we have glyceraldehyde 3-phosphate, each glyceraldehyde 3-phosphate produces 1 NADH and 2 substrate level phosphorylation. So if you have a look at it, in the first step 2 ATPs are 2 high energy bonds are lost. Then we're getting two NADH, one from the dihydroxyacetone phosphate and one from the glyceraldehyde 3-phosphate. So 2 NADH, that is 5 ATPs, and 4 substrate level phosphorylation. So if you add them together, -2 plus 4 plus 5, that is a net of 7 ATP molecules or high energy bonds are produced in each reaction, each step of each reaction of glycolysis. One glucose molecule yields 7 ATP molecules when it produces pyruvate via the glycolysis. The regulation of glycolysis. Regulation occurs by the key enzyme hexokinase, phosphofructokinase, and pyruvate kinase. Fructose 2,6-bisphosphate and fructose phosphofructokinase 2. They are two enzymes which also have a profound impact on the regulation of glycolysis. This will further elaborate when we discuss gluconeogenesis. Glycolysis is subjected to feedback inhibition by ATP, NADH, and citrate. ATP, NADH, and citrate are proxy markers to suggest that the cell's energy requirement is complete. Hence, they will inhibit the further occurrence of glycolysis, and the glucose 6-phosphate then could be utilized for any other metabolic pathways, or if the glucose 6-phosphate levels keeps increasing, they prevent further entry of glucose into the cells. Glycolysis is also regulated by covalent modification. All the enzymes in the glycolytic pathway are active in the dephosphorylated state. The moment a phosphate is put onto it by the protein kinase, these enzymes become inactive. Hormonal regulation. Insulin stimulates glycolysis, whereas glucagon inhibits glycolysis. Regulation at the genetic level is mediated by enhancement of expression of enzymes of glycolysis by insulin and its second messengers. The glucagon, on the other hand, inhibits the expression of these enzymes. I would be very happy to clarify any of your doubts, or if you feel free to communicate to me on my email. Thank you.